So today we are going to talk about movement of substances. So under movement of substances, they are being divided into two mechanisms. We have the passive transport and we have the active transport. In the passive transport, there is no use of ATP, that is energy. So it requires no energy for it to occur. In the active transport, it requires energy for it to occur. So under passive transport, we have two examples of it, or we have two types of it. We have diffusion and we have osmosis. So let's start with the passive transport. So let's start with diffusion. What is diffusion? Diffusion is the process through which molecules, these molecules, they can be in the liquid form or they can be in the gaseous form, or ions that move from the region of higher concentration, that means where we have more of it, to the region of lower concentration where we have less of it. Now, if I spray air freshener in this room I'm sitting, if I spray at this room from this point here, they will move to where they are not found, or they will move to where we have less of it. That means they are moving from higher concentration to lower concentration. But if the strengths have circulated throughout the room, it has become uniform, the diffusion will stop. That's why we have it. But the process stops when there is an equilibrium. That means when it has become uniformly distributed. So we have factors that affect diffusion. One is the surface area. As we say, the larger the surface area, the higher the rate of diffusion. For example, if we have two area, one area in which diffusion is occurring is very small, and the other one where the diffusion is occurring is large, what do you expect? Where it is large, we have high rate of diffusion to where we have just a small area in which diffusion is occurring. Example, in amoeba, it has a large surface area to volume ratio. For them, they don't need transport system to transport substances. But for us, we have we have a small surface area to volume ratio. In this case, we need transport system to transport the substances because diffusion alone will not be enough to do it since we have small surface area to volume ratio. So we talk about next, which is the size of the molecules. The smaller the molecules, the faster the rate of diffusion. For example, if I have a cube of salt like this, I drop it into a beaker containing water, and the other one I have the same cube, I smash it or I grind it and I put it into another beaker. The one that I have grinded will diffuse faster compared to this one that is in this cube form. So that tells us that the smaller the molecule, the faster the rate of diffusion. Next is the presence of concentration gradient. That means the difference between the two. So the higher the concentration of the molecule of a substance, the faster the rate of diffusion. For example, if this room was already having air freshener in here, then I spray, like the movement of those substances will be low because we already have it here and they move from region of higher to lower. But since the difference is very small, like someone may take a longer time even to realize that I have added or I have sprayed more. But let's imagine there was no air freshener in this room. Then I spray air freshener here. Since the difference over there is nothing there, they will move very fast to occupy those vacant space. So they tend to move very fast. But if air freshener was already spraying this room, then I spray, the movement will be very slow. Next, we have is temperature. The higher the temperature, the higher the rate of diffusion. So in the biology lab, most of I used to tell students that if the reaction is very slow, heat it. What do you experience? When you heat it, you see the reaction become fast. The molecule they start to move, they collide with one another, and with the wall of the test tube. So this temperature, this one is telling us that the higher the temperature, the higher the rate of diffusion, as we are talking about what movement of what substances, or even for the cook, those that used to cook, you can experience when the water boils, what happens. You see the water moving from one side of the pot to the other. That tells us also that the higher the temperature, the higher the rate of diffusion. And also the nature of the molecules involved. Molecule of gases, they diffuse faster than those of liquid. This one is clear. If you spray air freshener here, I'm going to use air freshener instead as an example, it will move faster. But if I pour water here, for it to move to the other part, it will take a long time for it to move. And also, liquid, they diffuse faster than solid. The fast solid, they just vibrate along a fixed point. And we have agitation, which is also known as stirring, increases the rate of diffusion. So if you put a cube of salt in a beaker and you put a cube of salt in another beaker, and the one you continue to stir it, you continue to turn it, you continue to the one that you are stirring, they will diffuse faster compared to the one that you just put it, you don't stir it and just leave it like that. Next we have is osmosis. Osmosis actually I can say is a form of diffusion because it is only talking about water molecules, while diffusion is talking about what? Liquid and gas. Also, they move just like we have in diffusion, they move from region of higher concentration to region of lower concentration. But in this case, there is a condition. It has to be what? Across a semi-permeable membrane. This one has to be present in the definition of osmosis until the molecules are evenly distributed, just like we have in diffusion. So let's talk about the importance of osmosis in humans. How important is osmosis to us? Just imagine we are talking about movement of water molecules. So everywhere movement of water is occurring in the body, it's been counted as, as an importance of osmosis to us. So first, let's talk about reabsorption of water in the tubules of the nephron. The nephron they are found in the kidney. They are the one that has to filter the blood to form urine. In certain cases, we may have a lot of water in the collecting duct. So what has to happen? They have to be reabsorbed into the blood. We don't want to lose excess of water. So the reabsorption of water in the tubules of the nephron is one important of osmosis to avoid the loss of excess water through urine. And also reabsorption of water from large intestine into the bloodstream. Also, when we take in food, we take in water. The body may not allow all of this water for us just to lose them in the feces. So what has to happen in the large intestine? Most of this water will be reabsorbed into the bloodstream. And also it gives turbidity to human cell. It makes it firm. Also, we talk about the importance of osmosis in plants, the opening and closing of the stomata. So in the opening and the closing of the stomata, it requires water. When the leaf is having excess of water, so those water in the neighboring cells closer to the guide cell that is guiding the stomata, the guide cell, they will move into the guide cell and the guide cell will become turgid and open up and the leaf will lose the excess water. In the other form, if the leaf is not having water, what will happen? The opposite will happen. Now the water will move from the guide cell into the neighboring cells surrounding the guide cell. And when the water moves from the guide cell into the neighboring cell, the guide cell will close. So in that case, the stomata will close to avoid loss of water since the leaf is not having a lot of water in it. And also, the next one we'll talk about absorption of water via the root hairs. The water is in the soil and it is being taken up by the plant specifically through the root hairs. So they take up that water through the process we call osmosis. and also it gives turgidity to plant cell. It make it turgid. That means it make it firm. So we have also factors that affect the rate of diffusion, just like temperature. I've explained it before. That in the lab most often we use to test students hit the experiment for you to increase the reaction, for the experiment to go faster, for us to complete it quickly. So when you heat it, what happens? The molecules they start to move faster. And also I talk about the boiling water. When water boils, what happens? You see the water moving from one side of the pot to the other side. And also we talk about concentration gradient. That means difference between the two. So if I have a beaker and I put a pig bladder to separate the two, one I put 4 cubes of salt and the other one I don't put anyone. Since the concentration gradient between the two is higher, what happens? Movement of substances, the water molecule move faster from where we have more to where we have less. That means they will move from where there is no cube of salt to where we have the 4 cube. That's what that will happen. And also we have the surface area, which I have explained before in diffusion, that the larger the surface area, the faster the movement of molecule across the semi-permeable membrane. So we have key terms that we are going to discuss, key osmotic terms. We talk about tonicity, which is talking about concentration of a solution. When we talk about tonicity, we are talking about the amount of solute in the solvent. If the amount of solute is higher in the solvent, we can say that solution is highly concentrated. But if the amount of solute in the solvent is less, we can say that concentration is what? Diluted, or we can say it is less concentrated. So under this, we have isotonic. In the isotonic, mind you, when we are talking about tonicity, we are talking more about the amount of solute in something. So the isotonic, iso means same or equal. So isotonic solution is a solution that has the same solute concentration as another solution with which it is being compared. And the next one, we have hypotonic solution. Hypo means below, and tonic is talking about solute. So hypotonic solution is a solution that has lower. Mind you, the hypo can also mean below or lower. So hypotonic solution is a solution which has a lower solute concentration than another solution with which it is being compared. And also we have hypertonic solution is a solution which has the hyper also means what? Higher. Tonic talking about solute. So since hyper mean higher, so hypotonic solution can be defined as a solution which has higher solute concentration than another solution with which it is being compared. Before we move to the next slide, we have something to discuss here. There is a diagram. We have what we call hypotonic solution. Let's say in a hypotonic solution, you place a plant cell or an animal cell, what will happen? Since it is hypotonic solution, that means it has less solute. And any solution having less solute means it has more water. Let me repeat it again. Any solution having less solute means it has more water. And any solution having more solute means it has less water. It is the opposite. Having less solute means you have more water. And having more solute means you have less water. So since it is hypotonic solution here, means it has less solute, that means it has more water. As we define osmosis, we talk about it that water will move from higher concentration to lower concentration, or high water potential to low water potential. So since we have more water here, this is an hypotonic solution, the water will move into the animal cell. So what will happen in the animal cell here, as the water moves, it will first become turgid, later it will burst. In the plant cell, the water will move, they will continue to move, it will just become turgid, it will not burst because it has cellulose cell wall. But in the animal cell, we don't have it. In the plant cell, because it has cellulose cell wall, it will not burst. And also let's talk about isotonic. If you place an animal cell or a plant cell in isotonic solution, what will happen? It will just be normal because the amount of water that will be moving out will be equals to the amount of water will be equal to the amount of water that is coming in. And also in the plant cell, the amount of water that will be going out will be equals to the amount of water that is coming in. Let's talk about the hypertonic solution here. That means this one. In the hypertonic solution, if you place an animal cell, if you place a plant cell, what will happen? Since it is hypertonic solution, means it has more solute. Hyper means higher, means having higher solute compared to the animal cell. So what will happen, as I said, anything having high amount of solute means it has less water. So that means outside is having less water and in the animal cell is having more water. So water move from where we have high water potential to low water potential. So water will move out. So what will happen? This one will shrivel, or we can say it shrink. It's just like you have a bucket of water. After drinking it from the plastic, what will happen? You can experience it. It will now like shrink. It will fold inside, like those that used to drink this craft in our country. Yeah? After drinking from that packet of water, what do you expect? It becomes shrinking also. It will no longer be firm. And also in the plant cell, what will happen? Water will move outside, just like I said in the case of animal, the water will move outside. In this case, when the water moves outside, what will happen? It will become plasmolized. In certain cases, even the cell membrane will move away from the cell wall. So we have other osmotic term which we talk about turbidity. Turbidity is simply talking about the swelling of a plant cell due to osmosis, making it to become firm. That means the plant cell take in more water. As it take in more water, it becomes firm. In that case, we say it becomes turgid. And this happens when the plant cell is placed in a hypotonic solution. We also have flaccidity and plasmolysis, which is almost the same quest, but slightly this one is having a higher degree than this one. Both they are due to loss of water molecule through osmosis. This one making it soft. But in here we talk about the cell wall pulling away from the cell membrane, which doesn't happen in flaccidity. In flaccidity, it's just the loss of water. The cell will become shrink, but the cell wall will not move away from the cell membrane. So the cell membrane will remain intact with the cell wall. They are not moving apart in the case of flaccidity. But in here, in the case of plasmolysis, what will happen? The cell wall pull away from the cell membrane, or we can say the cell membrane pull away from the cell wall. And the last one, the active transport, which we say it involves the use of what? Energy. So it is the movement of substances from the region of what? Lower concentration to a region of higher concentration. Previously, when we talk about osmosis, diffusion, we say from the region of higher concentration to a region of lower concentration. For active transport now, it has taken a different dimension. Movement of what? Molecule from the region of what? Lower concentration to higher concentration using energy from the cell. So these are some of the factors that affect active transport. We have temperature, which I have discussed it several times, and we have oxygen concentration. So let's talk about the oxygen concentration. Mind you, we need oxygen for production of energy in the body. The food we take in will be oxidized by the oxygen we take in during respiration. So that tells us since the food need to be oxidized to give us energy, That tells you that the more oxygen we have, the more energy will be produced, because in active transport, we need energy to carry on the active transport. Without energy, the active transport will not be able to carry on, as it requires energy for the process to occur. So, in a brief summary, here there is a table here explaining the diffusion, osmosis, and active transport that we have discussed. So, as we define all of them as movement of substances. So this one is a movement of molecules or ion, which can be liquid or gas. And in osmosis, just movement of water molecules. It cannot involve gas, just water molecules. And active transport movements of substances, it can be any substance. This one and this one, they move from region of higher concentration to region of lower concentration, and this one going opposite way is from lower to higher, and this one is done a concentration gradient from higher to lower, and this one here, it has to be across a partially permeable membrane. This condition must be present for osmosis to occur across partially permeable membrane. And also for active transport, energy is required. This condition must be fulfilled for active transport to carry on. And this one is going against a concentration gradient. That means from lower to higher. Against the concentration gradient means from lower to higher. In favor of the concentration gradient means from higher to lower.